Welcome to This Explainer. Today, we're tackling a bacterium that is an absolute master of survival and evasion. We're talking about Listeria monocytogenes. Now, if you're deep into veterinary microbiology, this is a pathogen you absolutely have to master. It's an incredibly resilient environmental survivor, but honestly, more than that, it's a cunning intracellular ninja. It literally thrives where other bacteria perish. It hides inside the very cells meant to destroy it, and it strikes with devastating precision across multiple animal species, and yep, humans too. Okay, let's dive into this. Here's our roadmap for today. We're going to start with the microscopic traits that make Listeria such a tough survivor, break down its intracellular pathogenesis, and explore the clinical disease, especially its infamous link to silage. After that, we'll put on our veterinary detective hats for laboratory diagnosis, covered treatment, and finally, zoom out to those absolutely crucial One Health implications. So let's jump right into Section 1 and meet this stealthy survivor. Here are the high-yield microbiological facts you need to have locked down for any exam. Listeria species are small, gram-positive coccobacillary rods. They are catalase-positive, oxidase-negative, facultative anaerobes. But here is its stealthy superpower, the thing that makes it so dangerous. It's extreme tolerance. It can survive temperatures ranging from a chilly 4 degrees Celsius all the way up to a hot 45 degrees. It also tolerates a massive pH range, anywhere from 5.5 to 9.6. Because it's an environmental saprophyte, it's just hanging out everywhere, widely distributed in herbage, water, and feces. Oh, and keep that characteristic tumbling motility at 25 degrees Celsius in the back of your mind. That's a classic diagnostic clue we'll be using later on the lab bench. Now, as diagnosticians, it's critical we can differentiate the really dangerous players from the harmless ones. The genus actually has 6 species, but only 3 are pathogenic, with L. monocytogenes obviously being the star of the show. If you look at the breakdown here, notice how L. monocytogenes shows a narrow zone of hemolysis on sheep blood agar and is positive on the CAMP test when plated with Staphylococcus aureus. It also produces acid from L. rhamnos, but not D. xylose. Compare that to L. ivanovi, another pathogen, which has strong hemolysis but is CAMP negative. And then you have L. innocua. Just like the name sounds, it's a harmless bystander. No hemolysis, CAMP negative. This kind of biochemical fingerprinting is exactly how we lock down a definitive diagnosis. Moving on to Section 2, let's look at invasion and intracellular pathogenesis. This is where the story turns into an absolute biological thriller. This sequence right here is why Listeria earns the title of intracellular ninja. Step 1. The bacteria use specific surface proteins called internalins. These act like molecular grappling hooks to adhere to and literally force their way into host cells, even non-phagocytic ones. Step 2. Once swallowed into a phagocytic vacuole, you'd normally think, okay, game over for the bacteria. Not Listeria. Virulent strains secrete a cytolytic toxin called Listeriosis. This toxin literally destroys the vacuole membrane, letting the bacteria bust out and escape into the safety of the host cell's cytoplasm. Step 3, it gets even crazier. It hijacks the host's own cellular microfilaments to create a tail-like structure, basically rocketing itself around the inside of the cell. And step 4, it smashes into the cell membrane, creating these pseudopod-like projections that push directly into adjacent, healthy cells. So what's the big takeaway here? By spreading directly from cell to cell, Listeria completely evades the host's humoral immune defenses. Circulating antibodies can't even touch it because it never goes outside. All right, Section 3. Let's connect this microscopic ninja to clinical disease and silage. This brilliantly illustrates the critical farm-level trigger for listeriosis. Outbreaks, especially in European countries, are notoriously seasonal, often striking silage-fed animals in late pregnancy. Why? Well, it all comes down to pH. In good quality silage, the acid from normal fermentation keeps the pH nice and low, which effectively inhibits listerial growth. But if oxygen gets into that pile and you end up with poor quality silage, the pH rises above 5.5. Because of Listeria's superpower, that wide pH and temperature tolerance we talked about, it begins replicating massively in those surface layers. We're talking up to 10 to the 7th colony-forming units per kilogram. Combine that heavily contaminated feed with the naturally decreased cell-mediated immunity of an animal in advanced pregnancy, and you literally have a recipe for an outbreak. When clinical disease actually strikes, it usually presents in one of these 2 main forms. And here is a really crucial clinical pearl for you. Usually, only one form appears in a single group of affected animals. You have the neural form, famously known as circling disease. This takes about 14 to 40 days to incubate. You're going to see dullness, literal circling, a head tilt, and a unilateral facial paralysis that leads to drooling. If you look at the brainstem histopathology later, you'll see microabscesses and perivascular cuffing. On the flip side, you have the septicaemic form. This one is rapid, just a 2 to 3 day incubation. It is a swift systemic infection that we most commonly see in very young, vulnerable animals like newborn piglets, lambs, and foals. Let's shift gears to Section 4, Veterinary Laboratory Diagnosis. This is the central question for any veterinary diagnostician, right? Listeria can look remarkably similar to other bacteria on primary isolation. We have to be absolutely meticulous about specimen selection. You want brain tissue for the neural form, liver or spleen for the septicaemic form, and cotyledons or fetal tissues for abortions. But there is one very special technique we need to use when we're trying to isolate it specifically from brain tissue. That technique is called cold enrichment. Remember earlier when we said Listeria can survive all the way down to 4 degrees Celsius? Well, we use that exact trait to our advantage in the lab. Other competing bacteria go dormant or just die off at refrigeration temperatures, but Listeria, it slowly keeps growing. By holding a suspension of the medulla in nutrient broth at 4 degrees and subculturing it weekly for up to 12 weeks, we can successfully isolate the pathogen even when it's present in incredibly low numbers in the brain tissue. It's a waiting game, but a highly effective one. So once we finally have our isolates, we systematically rule out the lookalikes. Listeria colonies look a heck of a lot like Streptococcus or Arcanobacterium pyogenes. But those two are Catalase negative, while Listeria is Catalase positive. Boom, easy distinction right there. Then we run the CAMP test with Staphylococcus aureus to confirm it's L. monocytogenes, and not that harmless L. innocua we mentioned earlier. We'll also incubate it in broth at 25 degrees Celsius to watch for that classic signature tumbling motility. Fun historical fact, in the old days they even used the Anton test, which involved inducing keratoconjunctivitis in a rabbit's eye to confirm virulence. Thankfully, we mostly rely on modern bench tests and chemoluminescent DNA probes today. Section 5. Let's talk about treatment and control strategies. First things first on the farm, you absolutely must discontinue feeding that poor quality silage to pregnant ruminants. Period. For actual treatment, systemic ampicillin or amoxicillin can work if you catch it in the very early stages of the septicaemic form. But for the neural form, the clinical response is often pretty poor, though prolonged high doses of penicillin combined with an aminoglycoside might help. So the really crucial point here is all about prevention. Now, you might think, hey, let's just vaccinate the herd. But read the underlying biology carefully. Killed vaccines simply do not work. Because Listeria spreads directly from cell to cell, remember our ninja evading a humoral response, you need a robust cell-mediated immune response to fight it off. Standard killed vaccines just don't stimulate that side of the immune system effectively enough. Finally, Section 6, One Health and Human Risk. We can't talk about Listeria without recognizing that this pathogen is a significant human public health threat. For veterinarians and farmers, directly handling infected material, especially aborted tissues, can cause localized papular lesions on your arms and hands. It is a genuine occupational hazard, so wear your gloves. For the general public, transmission is overwhelmingly foodborne. We're talking about raw milk, soft cheeses, coleslaw, and uncooked vegetables. And terrifyingly, because of its heat tolerance and the fact that it hides inside cells, it can sometimes even survive standard pasteurization processes. In a healthy adult, it might just cause a mild flu-like illness. But in pregnant women, just like in ruminants, it can cross the placenta and lead to devastating abortions. Furthermore, it is absolutely life-threatening for neonates, the elderly, and anyone who is immunosuppressed. So we've seen how Listeria monocytogenes uses extreme environmental tolerance, Listeriolysin, and hijacked cellular microfilaments to turn our own biology completely against us. It seamlessly bridges the gap between a farm's silage pit and a hospital's neonatal ward. It challenges our diagnostic skills, it laughs at standard killed vaccines, and it entirely bypasses humoral immunity. So I want to leave you with this provocative thought. If a stealthy survivor like this can thrive in extreme pH, hide away inside our own cells, and occasionally survive the literal heat of pasteurization, what other intracellular ninjas are waiting out there in the environment and our food chain? Keep studying, keep questioning the world around you, and always keep that One Health perspective in mind. Thanks for joining this explainer.